So today we're going to talk something about autonomic nervous system pharmacology. Okay, so let me write the topic here with brand new red pen. Autonomic nervous system pharmacology. So before going deep into this pharmacology, first let me describe some anatomy and physiology of the autonomic nervous system. So suppose this is our brain, and this is the spinal cord, okay, and suppose this is your sympathetic fibers, and this be your preganglionic fiber, and this is your postganglionic fiber. Sympathetic fiber, and let this be your parasympathetic fiber. Parasympathetic fiber. Okay, this is preganglionic fiber of the parasympathetic fiber, and this is the postganglionic fiber of the same parasympathetic fiber. Okay, now what I'm going to tell you is certain neurotransmitters are released from the neurons here. Okay, and these neurotransmitters transmit impulse through these preganglionic fibers in the form of axon potentials. And these axon potential, when it comes through the membrane of this neuron here at the presynaptic membrane, it releases some chemical substance, suppose acetylcholine. This acetylcholine, which is released in this presynaptic cleft, it is taken by this postsynaptic membrane. When this postsynaptic membrane takes this acetylcholine, it induces axon potential, and this axon potential is again transmitted through this fiber, and it comes at this point, and this point is called motor end plate. Suppose this is your muscle. Suppose this is your skeletal muscle, and when this axon potential comes up to here, this causes release of another chemical, the same, let it be acetylcholine here, and this axon potential causes release of the acetylcholine in here, and this acetylcholine is again taken by this muscle cell. And let me make this figure a bit bigger here. Suppose this is a cell. Okay, I magnified this here. Suppose this is your muscle cell, and this muscle cell, suppose this has a certain structure called receptor. Okay, this is the receptor present on the cell membrane of this muscle cell, and what this receptor does is this receptor receives the acetylcholine released from this nerve ending, and it goes here, and when the acetylcholine is received by the receptors, here occurs certain kind of transducer mechanism, and let me describe that transducer mechanism. Inside this cell there is a protein called Z protein. Okay, and this has alpha component, beta component, and gamma component. Okay, alpha, beta, and gamma component, and this protein is named as ZQ protein. This protein once this ACH is received by this receptor, this protein is hydrolyzed into different components. The alpha component comes out, and the beta component and gamma component, they get separated from each other. And this occurs with the energy that the cell takes from the GTP. GTP hydrolyzes into GDP, and the energy that comes from here is utilized to hydrolyze these different components. Now, the alpha components of this GQ protein goes to a special enzyme called phospholipase C. This alpha component of this G protein, this activates this phospholipase C enzyme. And this phospholipase C enzyme, it hydrolyzes phosphoinositol diphosphate, this compound. And it hydrolyzes, suppose this is a cutter. This phospholipase C hydrolyzes this PIP2 into two different compounds, that is inositol triphosphate and diacylglycerol. And this inositol triphosphate within this cell, it causes a release of the pre-calcium ions from the endoplasmic reticulum. Okay, if this is endoplasmic reticulum, it induces the release of the calcium from this endoplasmic reticulum. Okay, and these calcium ions, when get released inside the cell, then it causes or increases the positivity inside the cell. And this calcium, once it increases the positivity inside the cell changes from the electronegative nature into electropositive nature, and this means that cell is activated now. So up to now what I told is, suppose this is your brain, and from the brain a neurotransmitter is released, and this neurotransmitter is transmitted through this fiber in the form of action potential, and again this action potential causes the release of the neurotransmitter in the presynaptic cleft. Okay, and this postsynaptic membrane takes the ACH or acetylcholine, and this acetylcholine again induces the action potential through here, and again at the motorin plate the ACH is released and is uptaken by the muscle cells. And when this muscle cell receives the neurotransmitter through the receptor, this transducer mechanism goes inside. This GQ protein gets hydrolyzed into alpha, beta and gamma different components, and this alpha component goes and activates the phospholipase C enzyme, and this phospholipase C breaks down the PIP2 phosphoinositol diphosphate into IP3 and DAZ. Okay, and this inositol triphosphate releases the endoplasmic reticulum releases the calcium ion from the endoplasmic reticulum and causes the cell or increases the electropositivity of the cell. So this receptor, which receives the neurotransmitters released by this sympathetic fiber, I would say the parasympathetic fiber, because parasympathetic fibers use the acetylcholine as the neurotransmitters. Basically, okay, I want to repeat this again. Parasympathetic fibers use the acetylcholine as their sole neurotransmitters. And if I change this to parasympathetic, then let it be sympathetic. Okay, this is parasympathetic fiber side and this is sympathetic fiber side. So and this is the parasympathetic fibers releasing the acetylcholine, and these are the sympathetic fibers which release quite a bit different neurotransmitters than this. For the preganglionic fibers, it releases the same acetylcholine, okay, but for the postganglionic fibers, it releases another substance called adrenaline or even noradrenaline or even sometimes dopamine. But basically, for now, let us consider only adrenaline here, okay? So the sympathetic fibers releases the acetylcholine or uses the acetylcholine as the sole neurotransmitters for the preganglionic fibers. So this is the ganglion, two ganglions, okay? This is the preganglionic fiber and this is the postganglionic fibers. Okay, and if sympathetic fiber uses acetylcholine as the sole neurotransmitter in the preganglionic fibers, then the sole neurotransmitter in the postsynaptic or postganglionic fibers is adrenaline or noradrenaline or even dopamine. Now, when the neurotransmitter adrenaline is released by this postganglionic fiber, suppose here is also a motorin plate, and this motorin plate now is represented by a muscle cell. And let me make the same receptor here. And this adrenaline that's released by the sympathetic postganglionic fiber is taken by this receptor. And here also there is a protein called GI. This was GQ protein, and now this is your GI protein, okay? GI protein. I stands for inhibitory. I'll explain later. And this protein is again hydrolyzed in expense of the hydrolysis of GTP into GDP, and this gets separated into alpha, beta and gamma, and this alpha component of this protein goes and deactivates adenyl cyclase, deactivates adenyl cyclase. And this adenyl cyclase, you know what it does actually? The main function of adenyl cyclase is to convert the ATP into CAMP, cyclic adenosine monophosphate. So when this alpha component deactivates the adenyl cyclase, the rate of conversion of ATP into CAMP decreases. Okay, and as a result, the function of CAMP, or one function of CAMP is to release calcium or to increase the calcium concentration inside the cell. Since the rate of formation of the CAMP from the ATP is decreased, now the rate of release of calcium inside the cell also goes down. And you know what these gamma and beta components do? This also causes efflux of the calcium from the cell. Okay, now you see the concentration of the calcium inside the cell also goes on decreasing, and the potassium is also going out. So this cell is being more electronegative. In contrary to this cell was being electropositive. Okay, you can see now the difference between the cell. So this cell has been activated and this cell has been deactivated. Okay, this has been caused to be more electronegative and this has been more electropositive. So you see parasympathetic fibers release the acetylcholine or use the acetylcholine as the sole neurotransmitters in their both preganglionic fibers and the postganglionic fibers. And these neurotransmitters act on the cells of the effector organ through a special kind of molecules, and these molecules present on the surface of this cell through which the neurotransmitter act in order to modulate different functions of the cell is called receptor. And the receptor through which the parasympathetic nervous system acts are of different types, and the receptors through which the sympathetic nervous system acts are also of different types. So now let me introduce yourself with the different receptors used in sympathetic nervous system and parasympathetic nervous system. So you might know that autonomic nervous system basically consists of two parts. Okay, A and S consist of two parts: sympathetic and parasympathetic. So this parasympathetic nervous system uses acetylcholine as the sole neurotransmitters, and this sympathetic nervous system uses the catecholamines like adrenaline as the neurotransmitters. So these systems, sympathetic nervous system, is also called adrenergic nervous system. Adrenergic nervous system. And since this parasympathetic nervous system acts through the acetylcholine, this system is also called cholinergic. You can see choline and cholinergic similarity. Cholinergic nervous system. Okay.